Hi, welcome back. This is our second video presentation, and I want to discuss very briefly with you some of the core principles of genomics. So the ability to sequence genomes of organisms rapidly and cheaply has really changed the fundamental ways that we investigate the biology of organisms. It's become often the first way that we look at an organism, particularly for things like microbial species. So why do we, what are the different reasons that we sequence genomes? Well, the first of all, and one that's fairly easy to understand, is that we like to establish a catalog for an organism of the genes that exist. And in part, we do this because we're interested in understanding the kinds of functions that that organism might be able to perform in the environment. And so whether this is the ability of an organism to become a pathogen or to act as a pathogen, or its ability to metabolize certain aspects of its substrate, in part for those genes and the proteins that are produced by those genes that we understand their function, we can make a lot of inferences about that based on this. And we'll experience that later on in the class when we're looking at our own genomes. The other reason for sequencing a genome is really to establish a reference platform. So having the genome sequence allows you to do other kinds of functional assays on that particular organism. For example, once we've established the catalog of a gene, if we look at the RNA sequences that are produced during the development of that organism or in response to its exposure to a particular substrate, we can look at those RNA sequences and compare the amount of RNA that's in a sample for each of the genes and understand something about gene expression and variable gene expression. We can also use that platform in the sequencing of large numbers of individuals or samples from populations to understand sequence variation and how that varies across an environment or across samples. In that context, it's a very important and effective way to understand things like biodiversity and the ecosystem function that exists out in the environment. So we apply those basic principles to that by sequencing environmental DNA in a process that's referred to often as metagenomics. And then the development of sequences and the engagement in sequencing of genomes, whether they're people or other kinds of organisms, involves a lot of different ethical, legal, and social implications. So it becomes a very interesting process to think about in a broader context. So the most important thing to keep in mind that will become really important later on and help us focus our workshop is that there's a lot of variation in genome structure across different aspects of biology. On the other hand, the genomic DNA of essentially all organisms is really the same. It's a double-stranded DNA helix. And so with the exception of some viruses out there that have different uses of nucleic acids for their genomes of sorts, most organisms really, the DNA itself doesn't look any different. However, the genomes of organisms do vary extensively in things like size. So within a group of organisms, there's often very little correlation between the size of the genome and the number of genes that exist. And so this is something that's been referred to as the C-value enigma. And we'll talk about that a little bit more in a second. The larger genomes do tend to have a lot more copies of particular sequences like transposable elements or simple sequence repeats and genes that are duplicated. And so for the very large genomes, it's not necessarily that they have a larger number of unique genes. It's not a simple relationship like that, but they tend to have a lot more repetitive sequences. And as we're going to learn later on, that repetitive nature of these large genomes can represent some intrinsic challenges to sequencing those genomes. The genomes of eukaryotes are often diploid and contain allelic variation as well. So we understand, for example, like ourselves, many eukaryotes carry 2 copies of each kind of chromosome, one that they got typically from each of their parents, and those copies are often not identical, and so we have different alleles in each copy and produce something that we refer to as heterozygosity, which is also a challenge, but also a very interesting aspect of genomes that we can study by sequencing. So let's just look over some of the variation in genome size here. And so you can see that there are very, very small genomes. So for example, this very small, the smallest genome that exists is from a microbial organism that's actually in a relationship and functions together with leafhoppers, if I'm remembering correctly. And so you can see that genome is very small. It's only a little more than 100,000 base pairs, only has a little more than 100 genes, and you should really look that up. It's a really cool genome. It's not an independent organism in the sense that it doesn't live out in the environment by itself, but lives in association with an insect. And going from there, we increase genome size across a whole variety of different organisms. We'll talk about in a second. And you can find us down there toward the bottom, the mammals, where we're fairly tightly clustered around the typical mammalian genome size of about the same as ours, is about 3 billion base pairs, or gigabases. But we are by no means, although we consider ourselves very complex creatures, we are by no means the largest genomes. And so I've looked up and found this beautiful plant here that has a much larger genome size than ours, of 150 gigabases. And that genome, as you might expect, is filled with large numbers of repetitive stretches of DNA. It's very difficult to analyze. So one of the ways that we can think about genomes is think about gene density. And so that can go from very highly dense genome structures like our own. One of the genomes that we have in our cells is the mitochondrial genome. It encodes for a very small number of genes, but in a very small amount of space. And so, in fact, this is one of the most densely packed genomes that you'll ever come across. In fact, there are some cases where in the mitochondrial genomes, they save space by not even completing their protein coding aspects of the genes. They wait for processing later on, and include cases where genes are actually overlapping each other, which is fairly rare in biology still. And then we have our bacteria with 500 to 1,000 genes per million bases. One of my favorite creatures, the nematode C. elegans, has a little bit less dense genes at about 200 per megabase. But then when you look at ours, we only have about 12 to 15 genes on average, although these are not just randomly distributed genes per million bases of sequence. So if you look at this, one of the things that you will understand is that we are focusing our efforts on small genomes. So most of you, I suspect, will be looking at, in your projects, be looking at bacterial genes where the opportunity to observe and annotate large numbers of genes per megabase of sequencing effort will really pay off. So before we move on, I just want to emphasize the fact that bioinformatics, these algorithms that we'll experience later on in class, in this workshop, are really based on core understanding of cellular processes that are part of what you learn in cell biology, genetics, microbiology, and molecular biology classes, and biochemistry classes. When you think about it, when we put together DNA sequences and we look at them as just a big string of G's, A's, T's, and C's, a core goal of bioinformatics is to try to understand what do those strings of sequences do, what are they capable of doing in the cell. And really, that's done with computer algorithms that are just reverse engineering what we understand about the processes of transcription and how that works, and the rules of translation, both of which are, while not universal anymore, as we understand, those are nearly universal, and we understand when they aren't exactly the same in different organisms, which is often very interesting. And maybe one of you will run across something very interesting like that. So once again, thanks to New Hampshire INBRE, and we will see you in the next video presentation.